Good evening students. Today we will revise the reproduction 5 points. Number 1 point is the gonadal and genital differentiation. Gonadal differentiation occurs from bipotential gonads around 6th to 7th week of gestation. The cortex of the gonad give rise to the granulosa cell and oocyte. The medullary part give rise to the seminiferous tubules, Sertoli cells and Leydig cells. Then genital differentiation, we have two ducts. Okay, one is the Wolffian duct which gives rise to the male internal genitalia, and the Mullerian duct which gives rise to the female internal genitalia. This is the first point. The second point is spermatogenesis, the development and maturation of spermatozoa from spermatogonia. From one spermatogonia, 512 spermatids form, and hence 512 spermatozoa formed. It occurs in various stages. The duration is 70 to 74 days. From spermatogonia, the primary spermatocyte is formed, then secondary spermatocyte, then spermatid and spermatozoa. And the third point is menstrual cycle. Menstrual cycle, you have uterine changes, ovarian changes, vaginal changes and cervical changes. So ovarian changes is follicular phase that shows the development of follicles. From primordial follicle, primary follicle will be developed, which consists of layers of granulosa cell and oocyte. Then we have secondary follicle, which consists of theca layer, interna and externa, granulosa cell and oocyte. Then we have the tertiary follicle, which contains the antrum filled with fluid in between the granulosa cells. Then we have the mature Graafian follicle, which contains a single layer of granulosa cells surrounding the oocyte, that is corona radiata, and the attachment part is called cumulus oophrus. So this is regarding the follicular phase, how the primordial follicle develop into the mature Graafian follicle. The second is the ovulation. So once the mature follicle forms, a stigma is formed, a prominence is formed, and rupture takes place, and ova is released. The third is luteal phase. Now after the release, the Graafian follicle is converted into corpus hemorrhagicum. Then after around 22 days, it converted into albicans, corpus luteum, which is lipid laden Graafian follicle. Then around 24 days, it is 24th day, it is converted into a scar tissue called corpus albicans. So corpus luteum, corpus albicans. So if fertilization occurs, this corpus luteum stays back, and it will be persisted till 3rd month of pregnancy. So this is the changes in the ovary. Then changes in the uterine or menstrual phase. It is a shedding phase where the stratum functional shed off due to intense vasoconstriction under the influence of the local hormone prostaglandins, and due to the lack of the estrogen and progesterone. The next phase is the proliferatory phase. Due to the effect of estrogen, the proliferation of the epithelium takes place, and the endometrial thickness increases from 1 millimeter to 4 millimeter, and new vessels formed, blood vessels formed. Then we have the secretory phase where the endometrial glands become sawtooth. So sawtooth endometrial glands, and we have the neovascularization. Here also we have angiogenesis will be there, and secretions will be very thick. Okay, so then we go for the cervical changes. In cervical changes, in proliferatory phase, the secretion will be watery, and you can test the secretion in the slide. It gives a fern pattern, that is called fern test. In secretory phase, the secretions from the cervix will be very thick, okay, and tenacious secretion. And regarding the vagina, we have the squamation of the epithelium will be there, conification of the epithelium will be there in proliferatory phase. In secretory phase, it will be infiltrated with leukocytes. So these are the changes occurring in the female sexual cycle, which occurs every 28 days. And the usual menstrual blood volume is 30 ml. And menstrual blood does not clot because of the presence of the fibrinolysins. So this is the reason why menstrual blood does not clot. Then coming to the fourth point is the contraception. We have male contraceptive methods and female contraceptive methods. So contraceptive methods prevents the conception. So in male we have the temporary methods and permanent methods. So the temporary methods are the barrier methods, male contraceptive pills, and we have the natural method like coitus interruptus. And we have the permanent method in males, it is vasectomy. And there is no scalpel vasectomy, where an elastomer is injected into the vas deferens, and the vas deferens becomes hardened, thereby preventing the movement of the sperm. Coming to the female contraceptive methods, here also we have temporary method and permanent method. temporary method is the female barrier method, like vaginal diaphragm, cervical caps. Then we have the intrauterine contraceptive devices. Then we have the so these are the temporary methods. Then oral contraceptive pills. So all have advantages and disadvantages. In a permanent method, in females we go for the tubectomy. So the tube portion is cut and ligated, the cut ends are ligated, that is tubectomy. Then the last point is applied aspects. In applied aspects, you should know what is trisomy. So trisomy, if it is 3x, it is super females. XXY means it is Klinefelter syndrome, and trisomy 21 is called Down syndrome. Then cryptorchidism, that is undescended testis, is called cryptorchidism. Then increase in the menstruation is called menorrhea, and absence of menstruation is called amenorrhea. It may be primary or it may be secondary due to pregnancy. The most common cause is pregnancy. And increased frequency of cycles is called as polymenorrhea, and painful menstruation is called dysmenorrhea. So these are the 5 points covered in the reproduction. Thank you.